Good morning all. Today I am going to discuss about the topic carbohydrate metabolism. So we already know that carbohydrates are the polyhydroxy aldehydes or ketones. So what are all the metabolic pathways or takes place in carbohydrate metabolism? Before going to explain about carbohydrate metabolism, we should know that what is metabolism. So metabolism is a series of chemical reactions which are occurring in the living system. So there are mainly two types of metabolic reactions. The first one is catabolism and the second one is anabolism. So what is catabolism? Catabolism means it is the breakdown of complex molecule into simple one, that is catabolism, and with a concomitant release of energy. And next one, second one is anabolism. So anabolism is the opposite of catabolism. That means formation of complex molecules from the simple precursors. So there are mainly two types of metabolism. First one is catabolism and the second one is anabolism. Next is types of metabolic reactions. So for the metabolic reactions of carbohydrates, some reactions are takes place. So these are the different types of reactions which are takes place in the carbohydrate metabolism. So the first one is oxidation reduction reaction. So what is oxidation? Oxidation means addition of oxygen and reduction means addition of hydrogen. Second one is group transfer reaction. Group transfer reaction means transferring of functional group from one compound to another compound, that is group transfer reactions. And the third one is rearrangement and isomerization. So we already know that isomerization means the compounds having same molecular formula but different configuration. So that is isomerization reaction. And the fourth one is make and break of carbon-carbon bonds. That means sometimes making of double bonds. The single bond is converted into double bond and sometimes double bond is converted into triple bonds. Like that, break of carbon-carbon bonds means triple bond is converted into double bond and the double bond is converted into single bond. So these are the different types of chemical reactions which are taking place in the carbohydrate metabolism. Here we will see what are all the different types of carbohydrate metabolism. So these are the major pathways of carbohydrate metabolism. The first one is glycolysis. So glycolysis is also known as Embeden-Meyerhof pathway. That means it is the oxidation of glucose to pyruvate and lactate. That means the starting compound will be glucose and the glucose is converted into pyruvate and lactate under certain reactions. That is glycolysis pathway. Second one is citric acid cycle, which is also known as Krebs cycle or tricarboxylic acid cycle. That is the oxidation of acetyl CoA to carbon dioxide. So it is the final common oxidative pathway for carbohydrates, fats or amino acids through acetyl CoA. The third pathway is gluconeogenesis. So the gluconeogenesis is the opposite of glycolysis pathway. That means the synthesis of glucose from non-carbohydrate precursors. The examples are amino acids, glycerol, etc. So for the glycolysis pathway, the end product will be pyruvate or lactate. So in gluconeogenesis, the reverse reactions are takes place. That means the pyruvate is converted into glucose. That is gluconeogenesis. The fourth one is glycogenesis. So glycogenesis means formation of glycogen from glucose. That is glycogenesis. And the fifth pathway is glycogenolysis. Glycogenolysis means breakdown of glycogen into glucose. Next is hexose monophosphate shunt pathway or also known as pentose phosphate pathway or direct oxidative pathway. So this pathway is an alternative to glycolysis and TCA cycle for the oxidation of glucose. That means directly to carbon dioxide and water. So these are the 6 major metabolic pathways which are takes place in carbohydrates. First, we will see what is glycolysis pathway. So, glycolysis is derived from the Greek words, that means glycos is sweet or sugar, and lysis means dissolution. So basically glycolysis is defined as the sequence of reactions which is converting glucose or glycogen into pyruvate or lactate with the production of ATP. Next is, what are the salient features of glycolysis pathway? So the first salient feature is glycolysis takes place in all cells of the body. The enzymes of this pathway are present in the cytosomal fraction of the cell. So basically the enzymes for this pathway which is present in the cytosomal fraction. Second is, glycolysis occurs in the absence of oxygen. So in the absence of oxygen, the glycolysis is known as anaerobic glycolysis pathway, or in the presence of oxygen, that is also known as aerobic glycolysis pathway. So glycolysis takes place in two conditions. The first one is uh, presence of oxygen, that is also known as aerobic, and the second one is absence of oxygen, that is anaerobic. So for the anaerobic glycolysis pathway, the lactate is the end product, and in the aerobic condition pyruvate is the end product, which is then oxidized to carbon dioxide and water. Next salient feature, glycolysis is a major pathway for ATP synthesis in tissue-lacking mitochondria. So the tissue-lacking mitochondria means examples are erythrocytes, cornea, lens, so etc. So these are which does not having mitochondria. So in this time we have to synthesize or produce ATP through glycolysis pathway. Next salient feature, glycolysis is very essential for brain which is dependent on glucose for energy. The glucose in brain has to undergo glycolysis before it is oxidized to carbon dioxide and water. Next one is, glycolysis is a central metabolic pathway with many of its intermediates providing branch point to other pathways. So, the intermediates of glycolysis are useful for the synthesis of other biomolecules like amino acids and fats. Next salient feature is reversal of glycolysis along with the alternate arrangement at the irreversible steps will result in the synthesis of glucose. So that is the example of gluconeogenesis. Next, we will see what are all the main reactions of glycolysis pathway. So this pathway can be divided into three distinct phases. The first one is energy investment phase or preparatory phase. Second one is splitting phase, and the third one is energy generation phase. So these are the three stages which is occurring in the glycolysis pathway. Next, we will see what are all the reactions takes place in glycolysis pathway. So the first step is glucose is the starting compound. Glucose is converted into glucose 6-phosphate. That means glucose is phosphorylated to glucose 6-phosphate by the enzyme hexokinase or glucokinase. So these both enzymes are isoenzymes. So this is an irreversible reaction which is dependent on ATP and magnesium. The enzyme hexokinase is present in almost all the tissues. It catalyzes the phosphorylation of various hexose, that means hexose like fructose, mannose, etc. Okay, so the first step is glucose is converted into glucose 6-phosphate in the presence of hexokinase and glucokinase enzyme in the presence of ATP. So that is the first step. The second step is glucose 6-phosphate undergoes isomerization reaction to give fructose 6-phosphate in the presence of the enzyme phosphohexose isomerase in the presence of magnesium. That means, so we already know that isomerization means the compounds having same molecular formula but different in their configuration. So in glucose 6-phosphate, the functional group will be aldehyde group, and in case of fructose, the functional group will be ketone. So here the isomerization reaction takes place. The third step, that is fructose 6-phosphate, is phosphorylated to fructose 1,6-bisphosphate by phosphofructokinase enzyme. This is an irreversible and regulatory step in glycolysis pathway. That means the fructose 6-phosphate is again undergoes phosphorylation reaction. So in fructose 6-phosphate, the sixth carbon having phosphate group, and in case of fructose 1,6-bisphosphate, the phosphate groups are present in both first position, first carbon and the sixth carbon. So that is the third step. So these three steps are included in energy investment phase. The first step is glucose is phosphorylated to glucose 6-phosphate, and the second step is glucose 6-phosphate undergoes isomerization reaction to give fructose 6-phosphate, and the third reaction is fructose 6-phosphate is again undergoes phosphorylation reaction to form fructose 1,6-bisphosphate. Next is the splitting phase. So in splitting phase, the 6-carbon fructose 1,6-bisphosphate is split to two 3-carbon compounds, that is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase. That is the splitting phase. So and the enzyme phosphotriose isomerase catalyzes the reversible interconversion of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Thus, two molecules of glyceraldehyde 3-phosphate are obtained from one molecule of glucose. So that is the splitting phase. The third phase, that is energy generation phase. The energy generation phase starts from glyceraldehyde 3-phosphate. So the glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, and this step is important as it is involved in the formation of NADH and a high energy compound, that is 1,3-bisphosphoglycerate. That is the first step in energy generation phase. And the next step is 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate in the presence of phosphoglycerate kinase enzyme. That means the enzyme phosphoglycerate kinase acts on 1,3-bisphosphoglycerate, resulting in the synthesis of ATP and the formation of 3-phosphoglycerate. This step is good example of substrate level phosphorylation. Since the ATP is synthesized from the substrate without the involvement of electron transport chain. So these are the steps involved in this energy generation phase. The next step is conversion of 3-phosphoglycerate to 2-phosphoglycerate in the presence of phosphoglycerate mutase enzyme. So here the transferring of functional group from one carbon to another carbon is happening. That means in 3-phosphoglycerate the phosphate group is in third position, and in 2-phosphoglycerate the phosphate group is shifted to second position. That is conversion of 3-phosphoglycerate to 2-phosphoglycerate. The next step is 2-phosphoglycerate undergoes dehydration reaction to form phosphoenol pyruvate. That means removal of water molecule. So in 2-phosphoglycerate, the hydroxyl group from the third carbon and the hydrogen from the second carbon is removed as water molecule in the presence of enolase enzyme, and it will form phosphoenol pyruvate. That is the next step. The next step is conversion of phosphoenol pyruvate to pyruvate. That means the enzyme pyruvate kinase catalyzes the transfer of high energy phosphate from phosphoenol pyruvate to ADP, leading to the formation of ATP. This step is also a substrate level phosphorylation, so the reaction is irreversible. So that is the conversion of phosphoenol pyruvate to pyruvate. The next step is conversion of pyruvate to the conversion of enol form of pyruvate into the keto form of pyruvate. That means in enol form, the hydroxyl group is binding to the carbon-carbon double bond. That is enol form. So the enol form of pyruvate is converted into keto form of pyruvate. Next step is the last step. Pyruvate is converted into lactate. That means in the presence of lactate dehydrogenase enzyme, the pyruvate is converted into lactate by hydrogenation reaction. So that is the final step. The end product is lactate. Next is the energetics of glycolysis pathway. So there are mainly two types of glycolysis pathway, aerobic and anaerobic glycolysis pathway. So in aerobic glycolysis pathway, the end product is pyruvate. So how many ATPs are produced in aerobic glycolysis pathway? The total number is 8. How it's like so this is the entire glycolysis pathway. So in this glycolysis pathway, the splitting phase, that is uh, fructose 1,6 bisphosphate is splitted into two 3 carbon compounds. In that, the first compound is glyceraldehyde 3-phosphate. That glyceraldehyde 3-phosphate is converted into two molecules of 1,3-bisphosphoglycerate. So in that reaction, 2 NAD plus 2 molecules of NAD is converted into 2 molecules of NADH. That means 1 molecule of NAD plus means 2.5 ATPs are produced. So 2 NAD plus means, that means here 2.5 plus 2.5. The total number is 5 ATPs are produced. Like that, 2 molecules of 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. Here also 2 ATPs are produced. So 5 plus 2, 7. Then in the last step, that is 2 molecules of phosphoenol phosphate is converted into 2 molecules of pyruvate. Here also 2 ATPs are produced. So 5 plus 2, 7. 7 plus 2, 9. So total 9 ATPs are produced. In that 9 ATP, 1 ATP is utilized in the first step and the another ATP is utilized in the third step. So 2 ATPs are utilized in first and third step. So 9 minus 2, that means 7 ATPs. So in aerobic glycolysis pathway, the total number of ATP produced are 7 ATPs. So this is the number of ATPs synthesized in aerobic glycolysis pathway. So here we have mentioned the steps, each steps and number of ATP synthesized. So the first step is glyceraldehyde 3-phosphate dehydrogenase enzyme, which catalyzes to NADH. That means total 6 ATPs. And the second stage, phosphoglycerate kinase activate substrate level phosphorylation. Here the number of ATPs synthesized are 2. And the third step, that is pyruvate kinase enzyme, again activate substrate level phosphorylation. So the total number of ATPs are 2 and the 2 ATPs are consumed in the reactions catalyzed by hexokinase and phosphofructokinase. That is the first step and the third step. So minus 2. So the total number of ATP synthesized in aerobic condition is 7. Next is number of ATPs produced in anaerobic glycolysis pathway. So during anaerobic glycolysis pathway, one molecule of glucose is converted into 2 molecules of lactate. So here there is a net yield of 2 ATP. 2 molecules of ATPs are produced, and in the next step 4 molecules of ATP are synthesized by 2 substrate level phosphorylation. So total 4 ATPs are synthesized. In that 4 ATPs, 2 ATP molecules are used in first step and third step. Hence, the net yield is 2 ATP. That means 4 minus 2. Next is the significance of glycolysis pathway. So what are all the significance this glycolysis pathway? So the first significant is this pathway is utilized by all the cells and tissues to get energy, which is stored in the form of ATP and NADH. The second significant, it is employed in both aerobic and anaerobic respiration. So in aerobic condition, the end product is pyruvate, and in anaerobic condition, the end product is lactate. The process of glycolysis occurs in cytosol, so it is a very important process of energy generation for those organisms who do not possess mitochondria. The end product of glycolysis is pyruvate, which is an intermediate of various other processes such as gluconeogenesis, fatty acid synthesis, fermentation, etc. As we already know that gluconeogenesis means it is the conversion of pyruvate to glucose, that is the opposite of glycolysis reaction. Then next significant is even intermediates of the glycolysis process are utilized in other metabolic pathways. For example, DHAP, that means dihydroxyacetone phosphate, which is reduced to give glycerol 3-phosphate, which is used in the formation of triglycerides. Next is when there is a high demand of energy in muscles and there is an insufficient supply of oxygen, anaerobic glycolysis pathway is used to generate energy. Erythrocytes derive energy from lactic acid fermentation as they are devoid of mitochondria. Lens of the eye is another example of anaerobic glycolysis pathway. And the last significant, as most of the reactions are reversible, glucose is generated back from pyruvate by the process of gluconeogenesis. So these are all the significance of this glycolysis